Good. Okay. So good afternoon. My name is Liam. I'm here to talk about these lovely fish in the middle and to study these lovely fish. I work with these people, Greg Stevens, Josh Shavitz, Chiro Masai, and Tetsuo Izawa. And funding is from HFSP. And my slides are not moving. Oh, here we go. Okay, so before I talk about fighting in zebrafish, I want to talk about fighting in general. So what do we mean by fighting behavior? So we don't mean simply aggressive interactions between individuals. Throughout the animal kingdom, birds, mammals, you name it, try to assess each other. And they do it using a sequence of stereotyped interactions, which may or may not be aggressive or harmful to each other. In the language of game theory, there's a concept known as resource holding potential, which is some measure of the animal to, in broad sense, how good the animal is at life. So resources include food, mates, shelter, etc. So the idea when animals are fighting is that they're trying to mutually assess each other and get a sense of which one of them should, take the prize. In the models of game theory in particular, I'm going to come back to this idea of mutual assessment, where animals have physical contact, injuries are rare, and behaviors progress unidirectionally via phases, and we have escalation, while de-escalation, and I will make all these things a bit clearer as we move on. So before I talk about zebrafish fighting, I wanted to just show you a video of another animal and get a sense of what fighting is. So here we have giraffes fighting. So we start with displays, and they tend to look at each other and face off, and then we move into a kind of an aggressive shoving match where we're far away from any sort of injury yet. They're just doing things like assessing each other's mass and things, and it's entirely possible the fight stops here. But if they think they're, about the same, then bang, the fight starts, and they'll, fight until one of them gives up, or in, extreme cases, there is a serious injury or death. And so in zebrafish fighting on this side, we have a sketch of what a fight looks like from mythology. So the only points I want to make is that this is really a stereotyped sequence of multi-animal kind of interactions. And some of these behavioral states, which they describe here in this diagram, are kind of intrinsically multi-animal, such as chasing and fleeing, etc. So we want to, in a sense, just investigate this structure. So the zebrafish and how to entice them to fight. So a little background on zebrafish. They're a model organism for genetic, genomic, and developmental studies. A small fish, 2 to 4 centimeters in size, the adults typically. Because they have a sequenced genome, these genetic perturbation experiments are possible. And as a comment, they are very widely used in behavioral studies, but typically imaged in 2D. So how do you make zebrafish fight? If you simply put them in a tank, they're not going to start fighting necessarily. You have to treat them a certain way. So some of the things we've learned is that fighting, the probability of a fight occurring can be increased following a social isolation protocol. So we put fish in a situation where they're not, they're physically isolated, but not visually or chemically. So what this means is you have a transparent divider in the tank, stopping the fish from physically touching each other, but they can see each other and there is a flow of water. And if you take them then out of this social isolation and put them in a tank, there's a high probability they will fight, but there are also certain other conditions like they won't fight when they're expecting to be fed. At the bottom, we have a sneak peek at our experimental data, and this is a fish fight happening in our experiment. So how do you image 3D behavior of zebrafish or swimming animals in general? So we have this new experimental setup where we have a system with 3 orthogonal cameras for 3D tracking. The third is simply for redundancy. So we have 1024 by 1280 resolution. We film up to 150 hertz, and the tank is 40 centimeters cubed, much larger than the size of zebrafish. And lighting is backlit with LED panels. So one of the nice novel features of our experiment is that we have an optically transparent interior cage, which is designed to minimize reflection artifacts. So you can see without the cage, you have reflections at the side, you have reflections at the top, and without, or then with the interior cage, reflections are basically minimized or almost completely gone. So we've already seen sleep talked about once earlier today, and I'm just going to give a quick primer on it as well. So it is indeed a modified version of LEAP by Talmo Pereira, and he was going to give a talk at APS, and you can follow it to this session, which was going to be this morning, and I believe didn't happen. So what happens is we have, in effect, two networks, one for identifying body points. This is from the LEAP paper and follows the idea of LEAP or Deep Lab Cut. And if we look at the second figure, we see the problem when you move from a single animal to multiple animals, that identifying body points isn't enough. You have to be able to connect them. So the problem was solved first in the open pose paper using a concept they call part affinity fields, which is a 2D vector field joining body points. So you can think of this process as body points being nodes and the part affinity fields being the edges. And you learn not only where the body points are in an image, but how to connect them. In our current representation of the zebrafish, we're currently tracking 3 body points: the head point, the pectoral fin base, and the tail. So this isn't actually experimental data. This is just an illustration. Why 3 body points? It was a pragmatic choice at the start. We have plans to incorporate more body points. But you can think of this as a minimal model of a fish. You have a rigid bit at the front and tail moving about. And we make the point that we're working with Talmo to extend this tracking system that he's developing 2D in a 3D setting. So when we label our body points for our neural networks, we're using 3D information to place the body points. So why do we want to use body point tracking? So really we're trying to solve 2 separate problems here. One is we want a 3D postural representation for the fish, because we believe that for these interactions, the posture matters. Simply the centroid location won't give you, or you're certainly lacking information anyway. And we also want to maintain identity through complex occlusion events. So you can see on the left here a sequence of frames where glossing over details here, but basically we have 3 camera views, and we always crop where the fish are before we feed it to the networks, basically to minimize the pixel load. And what we're seeing here is that these interactions are very complicated, and from a purely image analysis point of view, it's a hard problem to, sometimes it's very hard to see anything that looks like a fish from any of the 3 camera views. And then this is an example of a static frame from the experimental data from the actual experiment. And we can see again, 3 cameras is kind of necessary. More cameras would be better. And on the right, we can just see a sample illustration of what we kind of get out. So 2 fish, 3 body points moving through 3D space. So once we have this data, which is in effect 2 fish and 3 body points in 3D for 18 degrees of freedom, we want to write down some coordinates to study these interactions. So the first thing you might think about is a fish-centric coordinate system for fish-centric behaviors. So we can use the vector from the pectoral point to the head of the fish to define an angle in the XY plane. And we can then, keeping Z the same as in the lab coordinates, define a Z prime and an X and a Y prime for these fish-centric coordinates. And we can see just an example of a fish swimming in these fish-centric coordinates where basically the fish is always pointing forward. And if you do PCA on this 5D representation, so just briefly why it's 5D. So we had 3 body points in 3D for 9 degrees of freedom. We want to make it translationally invariant. So we strip out 3 degrees of freedom and 1 more to make it invariant in the XY plane. And if you do PCA on this 5-dimensional space, you see that the most of the explanation of the variance comes from the action of the tail. And in particular, the dominant mode is associated with a tail motion in Z, which is nice because we're doing a 3D experiment and these are typically 2D experiments. 2 minutes. Okay, so now we have a pairwise coordinate system. So what we have is a fish-centric coordinate system but that lacks any sort of relative information between the fish. So we can define a second coordinate system and treat them like a pair. So we have relative coordinates and fish-centric coordinates. And our, one example really of a relative coordinate system is to, again, use a focal fish to define an origin and, for instance, write the second pectoral point or the pectoral point of the other fish in these coordinates. And we have a movie showing just an illustration of the dynamics where we've placed this in the PCA basis rather than in the lab coordinate basis. But the one point I want to make here again is that the mode explaining maximum variation or maximum variance is actually associated with a difference in relative Z height. Again, another kind of idea showing that a 3D experiment is important. So this is kind of early days in the project. It took a long time to get to this data, but now we have some ideas about going forward. We want to quantify this ritual fight. So there are ideas from game theory that we want to investigate. We have statements about the phasic nature of fights. For example, okay, you have some coordinates and you can look at the dynamics in these coordinates and cluster them using your favorite clustering method to get discrete states. Look at transitions between these states and see if these phasic structures really do exist. If there are subregions of the behavioral space that once you transition out of them, you don't go back to them. Current logic or current dogma is that you can't predict the winner of size-matched contests in these fish. So if you put two males in the tank, they will fight until suddenly they stop fighting, no indication that the end is coming. And we're working with an idea of a driven fish and a driving fish, and that the fight isn't actually symmetric, and we have initial early indications that it's not symmetric. And there are questions like, if you add more fish to the tank, 3 fish, will they fight? Are the interactions pairwise or are the triplet interactions? What's the optimal strategy for 3 fish, and do they follow that strategy? And I'll finish there. Thank you.
Okay, we have time for one question. Anybody raises their hand?
Oh, sorry, I think my own volume was muted there.
All right, I think we can just move on. Yeah. Okay. All right. So next we have Reed Panicasi Russo. Are you still with us?